I do not need the AI companion. Okay, all you.
Okay, thanks. So thanks for the introduction. So yeah, my name is Mayo Faulkner, and I'm a research software engineer at the International Brain Lab. And today I'm going to talk to you about how you can find the location of your recording electrodes in the brain. Okay, so basically this diagram shows the full workflow of all of the steps that you'll need to do pre-recording and post-recording in order to get an accurate reconstruction of where you recorded from the brain. So in the talk today, I'm just going to go through each of these different steps, go into some of the details and present to you some of the tools that are available to do each of these different processes. So the first one is probe labeling. So this happens prior to you doing the recording. So this is where you basically label your probe with a fluorescent dye. And the idea is that when you insert your NeuroPixel probe into the brain, it labels the proteins adjacent to the track, which you can then see once you image the brain later using a fluorescent microscope. So these shows a coronal slice and also a horizontal slice of a brain that has multiple neuropixels insertions, which have all been labeled with different types of dyes. So we have a red dye, which you can see here, a dye that fluoresces in the red, one that fluoresces in the far red or the purple, which you can see here, and also one that fluoresces in the green, which is much kind of a fine track here. So depending on your experiment, you would want to use either one or multiple of these types of dyes. So if you're targeting regions that are quite sparse and dispersed, then just using one type of dye will be sufficient. But if you're targeting regions like over multiple days and going into the same area, you might want to change up the type of dye so you can then accurately trace each dye depending on the day of the experiment. So you can see, for example, in this region here, the probes have been targeted in relatively close together in subsequent days. So you then want to change up the different dyes so you can accurately reconstruct your probes. So there are 3 dyes here that have been used. So the first is CM dye. So this is the one that fluoresces in red. And I think this is the one that's most commonly used by most experimenters. And this provides consistent labeling even, and yeah, there's low diffusion into the adjacent tissue. So this is a very nice dye to use. The second one is SB dialk. So this is the one that fluoresces in green. So this has very low diffusion tissue, but as you can see, it doesn't fluoresce so brightly. So you might need to adjust the amount of dye that you basically label onto your probe before you start. And the third is this DILC. So this fluoresces in the far red and purple. And this one diffuses more into the adjacent tissue. So if you're going to use this dye, it's best to use it toward the end of the experiment, close to when you're perfusing the animal, so that the dye doesn't spread too much into the adjacent tissue. Yeah, so once you insert your neuropixels into the brain, perform your recordings, and once you're finished with your experiments, you then sacrifice the animal, perfuse the brain, and fix it. And the next step is then to image that brain. So I'm going to go over some different 3D imaging techniques, but also some 2D imaging techniques just to highlight the fact that all of the subsequent steps, while it's more convenient to have a full 3D reconstruction, they can also be done with 2D slices. So the first 3D imaging technique is serial section 2 photon imaging. So this is what we've been using at the International Brain Lab to image hundreds of brains that we've been processing. So this basically is a 2 photon microscope connected to a vibrotome. And the idea is you take your perfused brain and you fix it into like an agar mount and you place it underneath the objective and you image the kind of coronal plane. And the vibrotome then comes in, takes away a section, exposes the next layer of the brain, and then you image the next coronal plane. And in this way, you work your way through the whole volume until you get a full 3D reconstruction of the whole brain. So in the IBL, we section at 50 micrometer intervals and then image in 2 planes for each section. So we then get a resolution in the AP stack of around 25 micrometers. So typically you image in 2, so green to pick up the autofluorescence of the brain and also red channel in order to pick up the fluorescent dye track. So this setup is relatively involved to set up at the beginning as it requires you having access to a 2 photon microscope. But once set up, it's relatively easy in that all you have to do is just suspend the brain in an agar solution and you can then place it underneath the objective. It does take quite a bit of time to collect the image, around 2 hours, as it slowly works its way through and slices through the brain. So the second 3D imaging technique is light sheet microscopy. So in this one, you need to first clear the brain. So you can see on the top right, we have a fixed brain and then after clearing. So basically that involves delipidating the brain in a solvent. And once it's adequately cleared, you can then put it into a light sheet microscope. And this basically, the laser is targeted perpendicular to the imaging objective. So here it's targeted in the coronal plane, and then you collect the image, and the laser works through the brain, and then you get a 3D reconstruction of the brain. Yeah, so this one has a lower resolution compared to the two-photon microscope, but for the purposes of track reconstruction, it's more than sufficient. It takes a little bit more preparation in that you have to clear the brain, and depending on the technique, this can take a couple of days to a couple of weeks. One advantage of this method is that you still have the full brain intact. So whereas this two-photon serial section, you slice away the whole brain, this one you still have your brain available if you want to then do further histological processing or immunohistology. The third imaging technique, 3D imaging technique, is the one I think is used at the Allen. So this is optical projection tomography. So similar to the light sheet, you need to clear the brain. The brain's then placed in this small chamber, and this chamber is then rotated at small angles, and an image is taken at multiple different angles. These projections are then all brought together in order to build a 3D image, and then you can then get a 3D image stack through the brain. This one is relatively, I think, cheap to set up, and it's available in open ephys. So I think if you're considering a 3D, maybe this is the one that you can set up most readily in the lab with a relatively cheap bill of materials. So I just also want to highlight that you can do this in 2D. So this would be a more traditional method where you take your brain, you find the sections that you're interested, use a vibrotome to cut the sections, mount them on a slide, and then just use a fluorescent microscope in order to image the slices that you're interested in. Okay, so once you have imaged your brain, the next step is to then register this to the Common Coordinate Framework. So this basically involves warping and moving your sample space or your image onto a template atlas in order that we can then assign a brain region to each of the voxels in our image. So what is the Common Coordinate Framework or Reference Atlas? So this is basically an average mouse brain that's been obtained by imaging over 1,600 different mice at the Allen Atlas. There's an averaged mouse brain, and each voxel within this brain has a brain region assigned to it. And in this way, you can do one-to-one lookup between 3D space, XYZ space in your image and your mouse volume to a brain region. So in the case of the mouse, it's this Allen Brain Atlas that's used, but in other species, there are also similar reference atlases that are available. So the idea behind this registration step is that you have your original image in your sample space. You have this template space, which is in this case, the mouse Allen Brain Atlas. And what you want to do is basically shift your sample space onto this template space. So this is typically done in 2 steps. The first is a linear affine transform, where you kind of broadly match the 2 images together. And then there's a nonlinear transform that deals with any little kind of small changes deformations that it needs to do in order to better match the template. And so the reason, why you want to do this is basically once your image is in the sample space, you can then do a one-to-one lookup to a brain region. And so you then know which areas in the brain you recorded from. So this shows an example of a 3D volume after registration. So on the left is the original volume, and on the right is after it's been registered to the Brain Atlas. And you can see the brain regions are overlaid, and you can see that there's basically a nice match between the brain region and boundaries and the actual brain, the fluorescence of the brain. So I should say that the registration is normally done on the green channel, so on the autofluorescence of the brain. And once you have that transform parameter, you can then apply it to the different channels that you've also recorded from, so the red channel or the blue channel, depending on what you imaged. So to do this in 3D, there's 2 packages, or there's more, but these are 2 that I'll mention today that are available. So the first is BrainReg, which is part of this brain globe architecture developed at the SWC. So this is a nice tool where it's like a desktop GUI that you can load in. You can load in your sample, your image volume, and then the target, and then it kind of just press run, and then it will do the registration for you, and it has some predefined registration parameters, and it will then align it to the Allen Brain Atlas. The second is Brain Register. So this is a Python command line tool, and And this one, you have basically more control of the parameters. So you pass in your sample volume, your template volume, and also parameter file. So if you want more control of how the fit is done, for example, if you find the fit is not sufficient and you want to tweak some of the parameters, then this is maybe the tool to use. So in 2D, this is a much more manual process. There are a number of tools available, and this is the kind of the process that you'll need to go through. So the idea is first selecting slices of interest. So these will be slices that you want to, that you have your recording and you want to be able to register to the CCF. So you then need to make sure that each of the slices is rotated correctly. You then need to find the corresponding slice in the CCF, the Allen Alice that most closely matches the slice that you're interested in. And at this point, you also might need to rotate the slice in the AP plane. So for example, in this image, you can see that the hippocampus is not completely symmetrical. And this is, for example, if you haven't cut your slice completely parallel to the brain direction. So once you've found the matching slice, there's then an automated registration process where it applies these like affine and also B-spline transform. And then there's an optional manual registration step where you can add kind of reference points and use these to match the different regions together. Yeah, so as you can see, the 2D is a much more involved process, but it's still possible to register 2D slices to the CCF. So here are some tools for doing this in 2D. So the first is AP Histology, which is what I got all of the images from the previous slide from. So this is a MATLAB-based tool and GUI and also command line, and it supports both automated and also manual registration steps. One thing is it only supports the mouse atlas. So if you're doing it in other atlas, other species, you'll need to use a different tool. The second is Herbs, which is a Python-based GUI, and this has similar kind of tools available, and it also supports multi-species. And there's also another one called Universal Probe Finder, which is a MATLAB-based GUI, and I think it extends on this AP histology and also allows you to do this in different species as well. Okay, so once you've now warped your brain onto the CCF, the next step is to then reconstruct your probe tracks. So this is basically, getting a 3D reconstruction of where your probe tracks were in the brain. So here we show just an example of this process. So there are 3 different mice brains, and we can see that we have, for each mouse, we have multiple recordings, and we can then get their location, 3D location in the brain. So this tends to be quite a manual process and involves basically loading in your image stack and finding the fluorescent track of interest that corresponds to the probe that you want to track, and then kind of scrolling through the stack, designating points along the stack, and then fitting a line through these. And in this way, you can then get the 3D coordinates of where your line is in the probe is in the volume. So this shows one of these tools where you can load in the different slices, and then there's a plugin GUI that allows you to kind of scroll through and click as you go along. For 2D slices, you do the similar thing. You kind of choose your starting slice that has the first, the tip of your probe, and then the end, and then you kind of draw a 3D line of best fit through these 2 coordinates. So once you have your line of best fit through your probe, you can then get the 3D position in space, and the next step is then to assign your electrodes onto this probe. So depending on the geometry of the kind of the probe that you used, you then distribute your electrodes along this probe track. So in this example here, I think it was just a NeuroPixel 1 probe. So the first electrode is around 200 micrometers from the probe tip, and then the electrodes are distributed at 20 micrometer intervals. And in this way, you can then distribute your electrodes on this probe track. And because we are now in this CCF reference space, each XYZ position corresponds to a voxel, which corresponds to a brain region. We then get a first estimate of where we recorded from in the brain. Okay, so some tools to do this tracing. So for 3D, again, this brain globe package has a brain globe segmentation plugin where you can load in the volume and do this tracing. And there's also another tool called Lasagna, which is what I showed before, that allows you to load in the volume and then click as you work your way through the volume. In 2D, all of the tools that I presented before that allow you to do the registration also support this. So as part of an extra step at the end, you can then assign the 3D coordinates of your probes, basically. So the final step is then basically to incorporate electrophysiology features to basically get a more accurate estimate of where you recorded from the brain. So there's two reasons, or a number of reasons why you want to do this. So the first is that because we've warped all of our brains onto this common coordinate reference, we've kind of taken out individual differences between subjects. So for example, there's different brain distributions, we've kind of removed that information. So we want to also account for that information. And the other reason is, as you can see, sometimes the fluorescent track might diffuse into the adjacent tissue. And so there might be inaccuracies in locating the probe tip. So we might need to offset our electrodes a little bit in the brain. So the idea behind this is that we incorporate the data that we've recorded on our probe, so the electrophysiology, and use kind of our knowledge of what we expect from a recording, and use this to kind of guide where we expect our electrodes to be in the brain. So this diagram shows an example. So on the left, we have our probe and the histology regions that it passes through. And on the right, we just show the firing rate that we record on the probe. And as you can see, we have this region here that's white that corresponds to an air gap in the brain. And based on our kind of knowledge of what we expect there to be very low activity on this region. So what we can do is basically place reference lines on both the histology and also the electrophysiology, and use these to shift the location of our electrodes in the brain. So what we want to do is be able to apply offsets of what we've just shown in this case, but also to be able to scale the electrodes along our probe to account for differences in brain sizes, but also scale in specific brain regions to account for different distributions of brain regions within subjects. So for this, we have a tool. So yeah, here are a number of different features that you can look for. So this is a table from this paper here, where they've outlined some very clear features in the brain that you can use to do this alignment. So examples are, for example, the hippocampus has very nice features such as high power spectral density and high firing rate. You can also use the brain surface. So for example, if your probe hasn't gone fully into the brain, there should be a clear distinction between the brain surface and inside the brain. And also fiber tracks and air gaps within the brain are also useful, as you expect there to be very low activity. So here's an example of kind of some of the features in the histology that you can use. So in this case, we're using the dentate gyrus. So this has two characteristics. One is that it's got a very high power in the LFP band. And the other is that it's got these two molecular layers where we expect there to be basically no very low activity. So what we have is our histology, and on the left we show the power spectral density. So we can see that we have this high power spectral density region. So we can assign that to the dentate gyrus. And similarly, if we look at the firing rate across the session, the recording session, we have these two distinct bands here where the firing rate decreases. And so we can assign these to the molecular layers of the dentate gyrus. So these are the kind of features that you'll use to then guide your alignment. So to do this, we built a tool where you can basically show a number of different electrophysiology features on the left-hand side, alongside the histology. So you can click through and see different features that are extracted from the raw data, but also the spike sorted data. And the idea is that you have the histology, and also on the right-hand side, you can see the location of your channels in the brain and where they are along the fluorescent track. So in this tool, we use the same concept of reference lines that I referred to before, and we can add them onto the display and move them independently on the electrophysiology and also the histology. We can then use these to move our electrodes. So in this case, in this example, we're using the boundary between hippocampus and thalamus to basically shift the location of our electrodes up in the brain. So you can add a number of different reference lines. And so in this case, the other one is going to be set to the high firing rate in the middle of hippocampus. And once you're happy with your fit, you can then upload, and it will then save the new location of your electrodes in the brain. And in this way, you've basically got a new estimate of where you recorded from the brain using the electrophysiology features that you have from the data. Okay. So tools to do this. So there's this ePhys alignment tool, which is the one that I just showed the video of. So this was developed in the International Brain Lab. And this is a Python-based GUI and allows you to show these different features, many different plots alongside the histology. At the moment, it only supports the mouse atlas, and you need to basically provide spike sorting data and also your kind of 3D location of your probe track in the brain in order to perform the alignment. There's also a MATLAB based version of this, a part of this universal probe finder. And this allows you to, as you're kind of getting an estimate of where your track is in the brain, it allows you to display some different features, and you can use these to adjust where your probe is in the brain. And this supports multiple species. Okay. Yeah, so basically that covers all of the steps that you need. So after you've completed all these steps, you should hopefully have an accurate estimate of where you recorded from the brain. And then once you know where your channels are in the brain, you can assign the neurons and the spikes that you recorded from to these channels, and then perform analysis based on the brain regions that you recorded from. Yeah, so I think in these slides, I've got links to all of the packages and different references that I use. So I think you'll have access to these, so you can check them out. And there's also these two other references that have lots of information about the different stages and more in-depth lectures into some of the different steps as well, particularly in like the registration and the 2D registration. Yeah, and with that, I'd like to thank you for your attention and thank funders for all of this work.
Questions?
Hi, can you hear me?
I can repeat the question
Ah.
if you can.
Do you do some quantitative checks, make sure, convince yourself that the local distortions and non-linear information didn't misplace the whole, because central space to the.
So the question is, what kind of quantitative checks can you do to make sure that your nonlinear registration step from your imaging to the atlas space didn't incorrectly distort your probe locations? Does that make sense, Maya?
Distort your probe locations. So I think one way is to kind of check the accuracy of the registration is you can apply the transform from your sample space to the template space, and then from your template space you can do the inverse transform again, and then if the two match closely, then you know that your transform is quite accurate. I don't know if that answered the question.
One thing that's maybe worth pointing out is that when you do the lasagna step of tracking the points along your track, you know, if the probe presumably went through the brain pretty close to straight, but of course, as you're pointing out, like when that registration happens, it might not be straight anymore. But you can track points that are along a non-straight line. So if the this mouse's brain in vivo was like warped weirdly relative to the atlas, you can still track the points where the probe truly did go. And those points along that line can be used in the subsequent step. Yeah.
And you know, there's a band, but you have a signature expected for all right.
Yeah. Question was, what's the a bank or a reference of the electrophysiological features that you might expect to see on different parts of the brain?
What's the bank?
What is there a reference data set or reference document that describes the electrophysiological features?
I think so. This paper here has a nice table which I presented before that has some of the common features that you can align to. I think there are some regions that are well defined. So, for example, the boundaries between the thalamus and the hippocampus, for example. But yeah, I think it's a question of looking at the literature, seeing what you expect in the brain regions that you're interested in, and then getting an eye for the different kind of boundaries that you see. Yeah, I think that's something that, I mean, in the IBL we'd also love to have is kind of this reference of what we should expect. But Yeah.
Yeah, I think if you're targeting a specific brain region, like the literature plus your own recordings are going to be your best guide of what to see. The IBL has a project called the Electrophysiological Brain Atlas, which we're working on actively and is really nearing some kind of release, where we're trying to incorporate the whole IBL data set, which is recordings across the whole brain. And one of the like things that we're doing with that is a decoder that tries to predict what brain region a particular channel is in based on its features. And so I think sometime in the future, we'll release both a paper and that atlas in a usable form. I think the goal is to incorporate it in this GUI that Maya's working on. Yeah. So, yeah.
Do you have any tricks and tips for working with multi-channel 2.0?
Multi-shank.
Yeah.
Yeah.
Sorry,
Yeah. So the question was about multi-shank 2.0 probes.
Particularly in the alignment step or
Are you
in,
specifically thinking about the last step there or the?
or like just in general, like how to successfully understand where your shanks has been and how to tell the differences between the shanks. Stuff like that.
yeah, so I think,
You go ahead.
yeah. So I think typically if you don't overexpress the dye, like each, because I think the separation is around 200 micrometers, you're still able to differentiate the individual shanks in the brain. So in that sense, you can then reconstruct each shank individually quite clearly with the probe tracing. Yeah, and then in the alignment step, so I think it's useful to be able to like visualize all 4 shanks at the same time because you know basically they're bound by the physical probe. So to make sure that you don't shift one crazily compared to the others. So that's like a new feature actually that we've added into the alignment tool, which will soon be released, but they're testing it now in Nick's lab. So this will be also something that helps that alignment process when you have multi-shank probes.
Yeah, and then there's also something that both the Allen Institute and we do slightly different versions of, which is if you're using the 4-shank probes and you're recording from like the deepest sites, that only gives you like a really small window into the electrophysiological features. And ideally, like to do this process well, you'd ideally like to be seeing like a long stretch of features along your probe so you can see, really where all the transitions between brain regions are. And so we do this thing where we like switch into a channel map that covers a deeper depth along all 4 shanks, but at sparser coverage, and record a few minutes of that to help us do the alignment step. And I think Josh, you guys do something where you sort of switch serially between different banks and then record briefly at a bunch of different banks and then sort of assemble that into something that can help you do the alignment step. So something like that, I think is a pretty good idea, depending on your target brain region. Depending on your target, you may feel like you don't need that, but for some, I think it's useful. Let's do one more question. Again, there will be lots of times for questions this afternoon. Let's go back there.
I think we are using the polymer and we are doing the programming. We have to split in the electrode in order to insert information, and we're using PEG stiffening. The problem is that there should be code with the dye before the PEG coating or after PEG coating.
Okay. Yeah, I'll ask Mayo. I'm not sure we're going to know the answer to that, but we could also just talk about it more later. So my other question was, they're using a different kind of probe that's not neuropixels, and it's a thing where the probe is very flexible. So you encase it in a stiffener that then like later dissolves in the brain. And right. And the question is whether you can coat the probe with the dye before or after you apply the stiffener to the probe outside the brain. Do you know the answer to that?
No. Yeah.
Yeah, I'm not sure either, but we can talk more about it maybe afterwards. Yeah. Okay, let's move on. Thanks so much, Mayo. Really appreciate it.
Thank you.